My name is Daryl Boness. This is the continuation of the Editor's Select Seminar Series for the Society for Marine Mammalogy. Our speaker will be Alicia Hendrix, who is a PhD student at the University of Washington. She's in their environmental toxicology program where she's doing her PhD. Alicia has a bachelor's degree in biology from Scripps College, not to be confused with Scripps Institute of Oceanography, but Scripps College is a small liberal arts school also in California. Alicia's research has her PhD research has principally dealt with threats to marine ecosystems and their impacts on wildlife and human health, but she also did some work before starting her PhD work, and these other projects involve physiological stress and adaptation mechanisms in barnacles along the west coast of the US, tracking coral reef bleaching in the Caribbean. And from that, she followed up with work in Central and South America, engaging in studies about invasive lionfish marine pathogens, and also studying about strategies for resilience. She's now back in the US and focusing on her PhD work, which involves algal-produced neurotoxins in more northerly environments. I don't imagine she's there in the colder periods, but she is, let's see. She's focusing on work in the Alaskan Arctic, specifically on the neurotoxins domoic acid and saxitoxins, which permeate the food webs in the Alaskan Arctic and subarctic, as well as many other parts of the world. The threat of these toxins are in the Arctic likely increasing because of increasing temperatures due to climate change and making conditions that are much more favorable to these toxic algal blooms. Tonight's talk is based on a paper in the volume 37, fourth issue of Marine Mammal Science. Her paper is open access, so it will be available to everyone, whether you're a member of the society or subscribing to the journal or not. And I'll put a link if there's, I think it looks like a link was just popping up in the chat. So if you want a link to the paper, just go to the chat. So Alicia's talk will be about results of work conducted on ice breeding seals in Alaska, samples of which were collected during 15 years of Alaska native harvesting of seals for subsistence. One of the things that I think is particularly interesting is Alicia's work has potentially important implications for Alaska native communities. And one of the things that's of particular interest to Alicia is her building of partnerships with these communities and giving them an opportunity to have more to say. So with that, I'm going to turn it over to Alicia, and let's hear what she has to tell us.
Great, thank you. Is this looking good? Thank you for that introduction. I want to start by thanking the organizers for inviting me to speak, and of course, thanking all of the audience members for coming to listen. It will be my privilege to present to you some work that I completed last year as a part of my PhD dissertation. This work explores the use of ice seals as sentinels for algal toxin presence in Alaska, and was completed in collaboration with researchers from multiple institutions. It really was very much a cooperative effort. I want to start by acknowledging that this work would not have been possible without the consent and collaboration of Alaska Native hunters and their communities, local governments, tribal councils, and the Ice Seal Committee. I want to specifically acknowledge here at the outset the contributions of the communities of Gamble, Hooper Bay, Kotzebue, Little Diomede, Nome, Point Hope, Point Lay, Safety Sound, Savunga, Shishmaref, Tunanak, Uktiagvik, and Wainwright. Again, this work really was a product, as you can see, of many, many people working together. So to start us moving through the research that this partnership allowed, I'm going to give you a quick outline of where we're going today. I'm coming to this work as a marine toxicologist, so I'm concerned with marine ecosystem threats and different environments' resilience in the face of these threats. Our focus today, and of my current dissertation work, is specifically the threat posed by harmful algal blooms. We'll talk about these broadly, the algal toxins that are the focus of this work, how wildlife can become exposed to them, and how exposures might be changing. Then I'll introduce our study subjects in the ecosystem where I'm studying HAB threats right now. I'll give you a brief outline of our collection and analysis strategy, and we'll get into the preliminary findings and what we can and can't say based on those findings. So as I mentioned, I study various threats to marine ecosystems. Many of the phenomena are well publicized, things like overfishing, coral bleaching, plastic pollution. These challenges all jeopardize marine environments, and marine environments that, when healthy, support human and wildlife health in myriad ways, including providing important protein sources, providing transportation options and trade resources, supporting tourism, many more ecosystem services. Increasingly, we're hearing about harmful algal blooms, which I will be abbreviating to HABs many times during this presentation. These bloom events occur when one or more algal species proliferate rapidly and produce large amounts of biomass. In some cases, algae subsequently synthesize a variety of toxic compounds with additional impacts on human and wildlife health. And there are multiple causative organisms and multiple potential toxins. I'm going to be focusing on 2 HAB toxins. The first of them is domoic acid, or DA, which is produced by diatoms in the genus Pseudonycha. And I'm showing an example photograph of these diatoms in the upper right. And domoic acid is an excitotoxin. What it does essentially is overexcite or overstimulate nerves, causing them to fire more than they should. There's a normal neurotransmitter called glutamate, and I've thrown the structure of glutamate up on the slide here. Glutamate, among other things, allows us to make movements. And you can see that glutamate and domoic acid have a similar portion of their chemical structures. This allows DA to bind to the same active site as glutamate. And then, in addition, some of the rest of DA's chemical structure means DA is more stiff than glutamate and binds harder, for lack of a better word. So this excess and persistent binding and subsequent activation of glutamate receptors by domoic acid causes a progression of symptoms, from GI distress through seizures into cell loss associated memory loss, and in severe cases, domoic acid can be lethal. We call this progression of symptoms, this acute toxicosis, amnesic shellfish poisoning, or ASP, because it's most common in humans following shellfish consumption. The second toxin, or technically suite of toxins that I'm going to be talking about, is saxitoxin, or STX. And these are produced by Alexandrium dinoflagellates, and there's a photo shown in the lower right. This group of toxins actually consists of several structurally similar compounds. They're generally referred to in units of saxitoxin equivalents. The chemical structure that's shown here is the sort of OG saxitoxin, and it and its analogs are able to bind to sodium channels. So if we think simply of DA as causing overexcitation, we can think of saxitoxin as the opposite. Saxitoxin blocks sodium channels, thereby blocking nerve function, so nerves can't fire. This causes similar GI symptoms to DA toxicosis initially, But as a poisoning worsens, we see things like paralysis instead of seizures. Lethality can also occur in saxitoxin poisonings, especially when we're talking about paralysis of the respiratory muscles. And the acute toxicosis associated with saxitoxin, is paralytic shellfish poisoning, the analog to amnesic shellfish poisoning for DA. So how do organisms get exposed to these toxins? Well, both domoic acid and saxitoxin move through ecosystems via food webs. They're ingested toxins. Once they're ingested, the toxins will move from the gut into the blood and in turn into the brain. So we have causative algae, Pseudonychia or Alexandrium, and they're shown here at the bottom of this simplified food web. Those algae are consumed by filter feeders, things like clams, which are in turn consumed by primary and secondary consumers. These might include seabirds, marine mammals, also humans. And these animals, particularly those at the higher trophic levels, can get doses of toxin that cause illness. So you can visualize domoic acid and saxitoxin sort of transferring along all of the lines representing consumption in this, again, simplified food web. So knowing that these algae can produce toxins, which can produce illness in multiple organisms, marine mammals and humans included, we of course start to ask questions like where, how often, to what degree are these toxic blooms occurring? And in the last 2 decades, almost half of the marine mammal unusual mortality events in the contiguous US have been attributable to algal toxin exposure. There's concern that wildlife exposure to these toxins may be increasing, along with rises in HAB frequency, duration, and geographic range. In 2015, associated with a warm water event, we saw the largest recorded Pseudonychia bloom in the coastal waters of North America. This bloom led to domoic acid-associated marine mammal strandings and deaths up and down the coast of the US. In this map, we're looking at the orange icons represent marine mammal strandings and the red icons represent marine mammal deaths. The bloom also caused closures of important shellfish fisheries up and down the coast. And those are shown in the shaded and hashed areas on the map. Importantly, there were poisonings of California sea lions reported in Long Beach, Washington, which was the first case of documented sea lion poisonings that far north. This has prompted researchers to speculate that warming waters may allow domoic acid blooms to occur in broader areas in the future. Saxitoxin incidence has also been increasing in frequency. Saxitoxin is known to occur further north than domoic acid generally. Recently, a group out of Canada published a report on recorded HAB events around the country. Now, this figure can be a little bit complicated, but we're looking at 3 maps. Each represents the same region on the west coast of Canada, and each map covers a consecutive 10-year period. So on the top, we have 1988 to 97, in the middle, 1998 to 2007, and on the bottom, 2008 to 2017. And the dots represent the number of years out of the 10 represented by each map that had documented saxitoxin events at each location. That means the dot in the 1988 to 97 window indicates that 5 of the 10 years in that period had reported saxitoxin events at that location. Similarly, the dots in the 2008 to 17 window indicate that all 10 years of that period had reported saxitoxin events at each of those locations. So if that's hard to follow, the overall picture here is that recorded events have noticeably increased in their frequency, their regularity, and the comprehensiveness within their range from the 80s and 90s to now. These saxitoxin bloom increases, like the 2015 domoic acid bloom, have also been connected to warm water events or trends. So if or when ocean waters warm, we see increasing incidence and severity of these types of harmful algal blooms. As you can probably anticipate, This raises concerns for areas where water temperatures are rising most rapidly. We know, for instance, that the Arctic and subarctic seas are undergoing rapid and dramatic changes in a number of ways. The most important to us in the context of this talk are warming conditions and loss of sea ice. This figure is courtesy Rick Tillman and the Alaska Center for Climate Assessment and Policy, and it shows the average sea surface temperature in the North Bering Sea over the last 120 years. You can see red dots showing the 10 warmest years and blue dots representing the 10 coldest. You can also see that all of the warmest years occurred in the last 2 decades, and the most recent coldest year was still about 45 years ago. This tells us that the problem of warming ocean waters is concerning and also that it's not going away. And one of the things that I in particular think when I look at this figure is that warmer waters and increased sunlight are things that promote algal growth. This likely means more HABs and more HAB toxins in these northerly regions moving forward. Now, the last thing I want to introduce us to before I get into the specifics of my project are some of the members of the Arctic and subarctic food web. So ice seals are so called because sea ice plays a critical role as a substrate for their pupping, nursing, molting, and other activities. And 4 ice seal species inhabit the Arctic and subarctic seas around Alaska. These are bearded seals, ringed seals, spotted seals, and ribbon seals. And they all participate in regional food webs as higher trophic level consumers. That means that they are candidates for HAB toxin exposures that we've been talking about. And it's worth noting that both bearded and ringed seals were listed as threatened under the Endangered Species Act in 2012. So additional environmental threats like HAB toxins are very worth investigating. So taking all of this information, the objective of our study was to quantify and assess temporal trends in domoic acid and saxitoxin prevalence in these 4 ice seal species in Alaskan seas between 2005 and 2019 in partnership with coastal Alaska Native communities that harvest ice seals for subsistence purposes. In other words, we're trying to answer the question, are toxins more prevalent over time and are toxin levels increasing? Are we seeing more toxin more often? And to assess this, we conducted an opportunistic study of gastrointestinal samples collected from subsistence harvested ice seals and subsequently shared with us. We restricted ourselves to samples collected in the spring and summer between the years of 2005 and 2019. And I'm showing you a map on the right here, on the right, with the locations from which we were given samples. So the various sampling locations are shown by black pins, and the classifications of these locations into regions are shown in the circles labeled with corresponding Alaskan seas. So these 3 seas, the Bering, Chukchi, and Beaufort, are the 3 regions into which we divided sampling sites and upon which we ran our analyses. Once we had the samples, we used an assay that's called an enzyme-linked immunosorbent assay to quantify domoic acid and saxitoxin in both samples. And we were then able to use statistical analyses to look for trends in both toxin prevalence over time via logistic regression and toxin levels over time in those samples where some DA or saxitoxin was detected via linear regression. So, what did we find? This is the interesting part. First off, we did detect both domoic acid and saxitoxin in samples from each of the Bering, Chukchi, and Beaufort seas. So here's that map of our locations, and I'm overlaying icons representing positive detections of toxin. Now we'll get into distinctions between the seal species later, but for now, we can note that icons in red represent detections of saxitoxin. Icons in yellow represent detections of domoic acid, and the icons in orange represent animals in which both toxins were detected simultaneously. And I want you to note that red, yellow, and orange icons occur in each of the 3 seas. So we know that both of the algal toxins are present at least sometimes and to some level in all three of these Arctic and subarctic regions. Now, how did toxin detections vary between the seal species? So it's worth noting out the gate here that our sample numbers varied by species. What I'm showing here are average sample numbers for each of our 4 species of seal. We did have slightly different sample numbers for each of the toxins. That's why these are averages. And the main thing to note is that our ribbon seals are significantly less sampled than our other species. Bearded seals represent the largest portion, but ringed and spotted seals are both relatively well represented too. So in that context, here are toxin detections by species. In the first row, you'll see the overall percentage of samples from each species that tested positive for domoic acid. And you can see that all 4 species had detectable domoic acid in at least one of their samples. In the second row, I'm showing the overall percentage of samples from each species that tested positive for saxitoxin. And you can see that bearded, ringed, and spotted seals all had some detectable saxitoxin, but no ribbon seals did. So right off the bat, we see that all 4 seal species are experiencing some level of toxin exposure. I also want to highlight here that bearded seals had the highest prevalence of both domoic acid and saxitoxin. Also, ringed seals had the second highest prevalence of both domoic acid and saxitoxin. And I'm flagging that now because we'll return to it later. We can also look quickly at some of the levels of toxin that we're finding in these samples that do test positive. So here I'm showing you just the maximum toxin levels observed in each of our seal species. That means that, for example, of all of those samples from bearded seals that tested positive for domoic acid, the highest concentration of DA in any single one of them was about 1,500 nanograms per gram, or 1.5 parts per million. Now, these maximum toxin levels obviously don't communicate all of the nuance and range of concentrations observed in each species, and they're also impacted by the number of toxin-positive samples from each species, but they can give us a snapshot and serve as just one more indicator in the overall picture of toxin exposures in this region that we're developing. And we can see that these maximum toxin levels echo the prevalence trends that we observed. Bearded seals, in addition to having those largest percentages of samples with some level of saxitoxin, also had the sample with the highest overall observed concentration of saxitoxin. Ringed seals, in addition to having the second largest percentage of domoic acid positive samples, also had the sample with the highest overall observed concentration of domoic acid. Now, on the map here, we do have icons representing each species for those toxin detections. It's a little hard to see here, and it gets into the weeds a bit, honestly, but I'd encourage you to take a look at our paper if you want to see more of those details. Now, as I mentioned, our cross-species comparisons are complicated by the fact that we did have different sample sizes between species. Again, our samples were opportunistically collected. These comparisons are also complicated by the fact that we had different proportions of stomach and colon content samples from each species. As it turns out, samples taken from different locations in the GI tract, even simultaneously, can report different toxin concentrations. Colon content samples tend to have higher toxin concentrations. That said, I still want us to think for a minute about what else might underlie those big difference in toxin prevalences between species. So you'll remember that both domoic acid and saxitoxin are ingested toxins moving through food webs. So it makes sense that there might be some relationship with what these seals are eating. Now, this is a simplified summary. The pictorial depictions are neither comprehensive nor are they perfect, but they're here to give you an idea of trends in prey preference. Bearded seals, which had the highest percentages of domoic acid and saxitoxin positive samples, also have the highest proportion of benthic organisms in their diet. They're actually primarily feeding on benthic fish and invertebrates, things like bivalves, annelids and polychaetes, amphipods, sculpin, that kind of thing. Ringed seals, which again had the second highest prevalence of both toxins and did have a high maximum DA level, eat mostly pelagic fish and invertebrates, but benthic fish like sculpin and inverts do show up in their diets as well. Also, the pelagic fish that they are eating include planktivorous fish like rainbow smoots. Spotted and ribbon seals, which had the lowest toxin prevalence and levels, eat pretty much exclusively pelagic fish, with reports of ribbon seals eating octopus and shrimp as well. They also consume larger proportions of cod, herring, pollock, and other fish that are either piscivores or at least consume some fish themselves. Now, this is speaking broadly, but what our data suggest is that in general, benthic organisms seem to be very potent. They appear to be more toxic vectors. Also, prey items that are feeding further down the food chain themselves, so fish that are eating algae directly as opposed to fish that are eating other fish, might be carrying higher levels of toxin to their predators. And again, we do go into a bit more detail about this in the paper, so I'll direct you there if you want to really get into it. But these are our broad conclusions looking at seal diets and possible correlations with toxin prevalence that we observed in our 4 species. So I'm going to turn now to the regression analyses that we ran over our temporal data sets. So these were looking for trends in toxin prevalence and levels over time for our seal species in each location. I'm going to start with our logistic regression results, and these pertain to the proportion of samples that tested positive for toxin, regardless of what actual quantified concentration of toxin was. And we did run these analyses on all of our seals at all locations from which samples were collected, or all regions from which samples are collected. But in many cases, our samples, again, because they were opportunistic, not a predetermined sampling regimen, weren't well suited to temporal analyses. We either had samples from different GI tract locations in different regions, or the years represented in our sampling data sets were inconsistent. So I won't be showing those results today. Our largest data set, that data set from bearded seals, was the best suited to these analyses. We had samples of the same type, stomach contents, in relatively equal numbers from both the Bering and Chukchi seas. And they covered a pretty impressive timescale from 2012 to 2019, relatively consistently year to year. So most of the years are represented at each location. This allowed us to compare trends between regions. We did have some bearded seal samples from the Beaufort Sea, but they tended to be fecal samples and were therefore not comparable. That's why they're not being highlighted here. So I'll walk you through this figure showing results from, again, stomach content samples from bearded seals in the Bering and Chukchi seas. We're looking at DA on the top, we're looking at saxitoxin on the bottom, the Bering Sea is on the left, and the Chukchi Sea is on the right. And you're looking at years along the x-axis, and then on the y-axis, we see the proportion of samples taken in each year that tested positive for toxin. Those numbers next to the data points are the sample size from each year. The overlaid blue lines with the confidence intervals are the results of our logistic regression modeling for each region and toxin. So these show the modeled likelihood that a sample taken in a given year would test positive for each toxin respectively. And this temporal trend for increasing domoic acid prevalence in the Bering Sea was the only significant trend. This means that the likelihood that a sample taken from a bearded seal in the Bering Sea would test positive for DA appears to increase from year to consecutive year. I mentioned earlier that we ran linear regressions. Those were looking for trends in toxin levels over time. So this is in contrast to the logistic regression for toxin prevalence, which we just looked at. For this, we used just our data from samples that did test positive for toxin, and we modeled quantified DA and saxitoxin concentrations from year to year. and we did not see evidence for significant trends in toxin levels. It's worth noting here that even though we had those maximum toxin levels that I reported several slides ago, the majority of toxin hits for these seals were low levels. And this probably is why no evidence for trends emerged. When most of the numbers going into a regression are low, the line you'll see stays pretty flat. So before talking a bit about what this means for ice seals, I'm going to pause and summarize our study's primary findings. First, we found domoic acid and saxitoxin in at least some samples from all three of the Arctic and subarctic seas. Second, domoic acid was found in some samples from all 4 species of seal, and saxitoxin was found in 3 species. Together, these 2 pieces of information suggest to us that ice seals are regularly exposed to domoic acid and saxitoxin in the Bering, Chukchi, and Beaufort Seas. Third, we saw that bearded seals and ring seals appear to have the highest prevalence and toxin levels, which is likely due in part to their different diets. Fourth, there is an increasing trend in domoic acid prevalence in the Bering Sea. That is, our logistic regression indicated that the likelihood of a Bering seal sample, of a bearded seal sample collected in the Bering Sea would test positive for DA increases over time. Now, this last statement is consistent with the idea of potential northward expansion of Pseudonychus species as waters warm. To be very clear, this specific project does not address this issue of range expansion directly, but our findings are consistent with this hypothesis. Now, all of these results so far are using samples from ice seals to indicate to us overall toxin and food web trends in the Arctic and subarctic. These seals are acting as sentinels for environmental toxin exposure conditions for everyone. I'm going to turn now to what these exposure risks might mean for ice seals themselves. Are toxin exposures high enough to impact the health of these ice seals? And the assessment of this is a bit tricky. We do not have very detailed data regarding the levels of toxin associated with acute toxicosis in marine mammals. We do have data like these, which show the domoic acid levels quantified in fecal and stomach content samples taken from California sea lions stranded with acute DA toxicosis on the California coast. So these are describing gastrointestinal tract levels reported in animals during a bloom event. You can see that the level of toxin in the sea lion GI tracts varies widely. And this is for a few reasons, most saliently that sampling occurs an unknown period of time after ingestion of contaminated prey. And sampling might occur after some ejection of matter from the GI tract, for instance, following toxicosis-related vomiting. But the data illustrate, or I'm hoping that they do, that diagnosis of acute toxicosis requires the observations of overt signs of poisoning, for instance, head weaving, seizures, ataxia, in addition to any measured toxin levels. So to that point, we can appreciate that hunters did not report any clinical signs of domoic acid-induced excitotoxicity, nor of saxitoxin-induced paralysis in our seals. So that's a good sign. Another thing that we can look to as we try to gauge health risks for seals are the human public health guidelines currently in place. So even though we don't have specific information on uptake and susceptibility to pathology for marine mammals, in lieu of that, we can look to what are thought to be protective levels for humans. Health Canada and the US Food and Drug Administration have both adopted regulatory limits for domoic acid in seafood of 20 micrograms per gram or parts per million, and regulatory limits for saxitoxin in seafood of 0.8 micrograms per gram or 0.8 parts per million. I'm relisting the maximum toxin levels that we observed in samples from any of our seals converted to parts per million. And you can see that they are comfortably below the human public health limits. Given these two pieces of information, then, the data that we've published don't show evidence for health impacts to ice seals at this point for either toxin. That said, though algal toxins may not yet be a significant health threat to ice seals, there are valid concerns about future exposure risks with continued ocean warming, because warmer ocean temperatures foster increased harmful algal growth, and Arctic and subarctic regions are undergoing rapid rates of ocean warming. Concern for increasing impacts of harmful algal toxins on important marine resources is high. This is also particularly important for communities that depend on these resources for a number of cultural and other reasons. We expect further that these harmful algal toxin exposure risks will increase. So with that, I want to thank my many collaborators without whom none of this would have been possible. That includes, again, the communities of Granville, Hooper Bay, Kotzebue, Little Diomede, Nome, Point Hope, Point Lay, Safety Sound, Savunga, Shishmarith, Tunanak, Uktiagvik, and Wainwright. It also includes my co-authors and the members of their many labs, as well as all of our funding sources. And with that, I'm very excited to take some questions and to hear what we're all thinking about.
Alicia, thank you for that very interesting talk. At the moment, I'm not sure if we have any questions lined up in the Q&A. Eric will be monitoring that for questions, but one question that came to me during your talk is to what extent we might know something about ocean temperature levels and the level at which saxitoxins and domoic acid actually becomes of concern and a clinical problem for marine mammals or people.
This is a great question. It's pertinent to the expansion of these algal toxin ranges. And there's a lot of research going on in many labs right now looking at the specific conditions that either cause Pseudonetia or Alexandrium to proliferate, and also conditions that cause them to produce toxin, because those don't always happen at the same time. And there are many thoughts on this. A lot of them are temperature related. Temperature does seem to increase toxicity and algal levels. There's also a lot of evidence that other stress conditions, so certain nutrient limitations, a variety of stress conditions, will also produce more toxin from algae. So it's a sort of complicated interplay of factors and an ongoing area of research to find out what the sort of suite of factors interacting will do in terms of algal levels in different areas.
Eric, I'll turn it over to you. I see some questions coming in.
Sounds good. Thank you for the wonderful talk, Alicia. And we have two questions here right now. I'll start with the first one from Lori Polasek. Thank you for your presentation. Very informative. I may have missed this earlier. Is there a half-life for either of the toxins, or once they are in the systems, are they permanent?
So I'm going to think about this on the organism level, because that's kind of what I do. And different organisms, different animals will take in the toxin at different rates, and they'll also depurate the toxin at different rates. So for instance, there was some work down in Monterey Bay back in the early 2000s that when there was a domoic acid bloom, they looked at levels in anchovies and sardines, and anchovies consistently had more toxin level than sardines did. So first thing to bear in mind is that when the toxins permeate a food web, they are doing it a little differently within that food web. And then second thing to think about is that the toxins stay in tissues for different amounts of time between animals. So in Washington State, where I'm based, razor clams are one of the most potent toxin vectors. Domoic acid stays in razor clams for a long time. It depurates very slowly. So there are other shellfish that might be exposed to the same toxin, but they won't hold on to it for as long. The toxins are also, there are systems for, I don't know, eventual breakdown and recycling out of the system. I think more so than the persistence of the toxin in the water, I think about the persistence of the toxin in the organisms that are interacting within the system, because that's how we really see toxicosis and toxicity manifesting.
Great answer. Next question from Anonymous. Do DA or STX bioaccumulate in higher levels of food webs?
So this is a related question. I like how we're thinking about this. When I think about toxins going up food webs, again, it kind of depends on the organism. The biggest thing for me is thinking about the overall trophic level. So if you have a consumer that's eating lots of organisms that have algae in them, that consumer can have high toxin level exposure. Domoic acid does go out of the system. It's usually on the scale of, well, we don't have great data specifically for marine mammals looking at how long the toxin stays in a marine mammal. But in, for instance, rodent studies, it's on the scale of days. That doesn't take into account in situ environmental exposures where as toxin is depurated, you might be bringing in new toxin as well. So again, this is a bit complicated, but I'm sorry, I lost my train of thought there. I need to loop back in, to be honest.
It's all good. I think you answered it. So there's a lot of complexities in how these things that there was the DA and the STX bioaccumulate.
Yes,
at
the
higher food webs.
bioaccumulation.
That was the core. Yeah.
Bioaccumulation is not like a huge concern for these toxins, but the exposures are complicated and the environmental exposures depend on a lot of things.
Yeah. I think you answered that sufficiently. Okay, next question from Michael Castellini. Is there information about how the toxins might distribute in different body tissues? I am thinking about harvest of species by local communities that may consume different organs.
Yeah, this is a great question. So when we look at, for instance, fish species, there is some data on the fish species, more so than any specific data on marine mammals. The bolus of the toxin stays in the GI tract. Domoic acid, for instance, is actually taken up at a pretty low rate into the actual tissues. So if you look at fish GI tract or whole fish versus fish fillet, the fillet has significantly lower levels of toxin. And this actually has implications for regulatory limits because, as was pointed out, and I love that this was rolled into the question, depending on cultural or other differences in how different animals are consumed or which parts of animals are consumed, limits and guidelines that we put into place to protect people from, for instance, fillets, maybe wouldn't protect people from who were eating specific organs. So thinking about where the toxins go in the system is critical to making good public health recommendations and considerations. Again, the toxicokinetics of these toxins, so where do they go in the body, how quickly do they do that, and how long do they stay there before they're metabolized or excreted? There's not a ton of research for marine mammals specifically. There is research in rodent models, but it looks a little different in those models versus other animals.
Thank you. And great point. I hadn't really thought about different organs and consumption of different parts of the animals, but it's a really good point in terms of the concentrations.
Yeah, thank you. Thank you to the asker of that question. That was a good observation.
Okay, next question here from Jonathan Tempty. Any thoughts on the potential effects of age of seals as newly weaned pups with lower body mass may have different preferences than adults? Was there any thoughts on how the potential effects of age of seals as newly weaned pups? So when newly weaned pups with lower body mass, I guess, will they have different food preferences than adults? Yeah,
Yeah,
question's a little layered, but
this is an exciting question, both from a seal perspective and also in the context of the rest of my dissertation. So first of all, there could be differences in the toxicokinetics of how things would move through mammals at different life stages. There's actually quite a lot of research that looks at in utero and developmental exposures to toxins in laboratory models. I've done some work showing how toxins can move to amniotic or allantoic fluid and possibly expose things in utero. Once you have a neonate or a young pup, they will be eating different things. So to me, the biggest difference in toxin exposure would come with differences in diet and feeding preferences. We do know that domoic acid, at least, does make it into milk. And so animals consuming mother's milk would still be exposed to domoic acid. I will say that we did look at the ages of our animals, and for all 4 of the seals, for those that we did have age data, we had representation of some pups, some subadults, and some adult seals in all of our data sets. Also, since we're on the subject of age, and I think about this a lot, I want to throw this out there. The next chapter of my thesis is looking at the association between toxicosis susceptibility and advanced age. So there's some evidence that, at least in the sort of canonical 1987 poisoning event where we recognized domoic acid as a human neurotoxin, there was some evidence that advanced age might be associated with increased susceptibility. And the second chapter of my thesis follows up on that, and it looks at that in a rodent model. So the other component of age that wasn't specifically asked in the question, but does come up for me, is in the more aged seals, particularly those who might develop underlying conditions that might change some of those depuration rates. I might be concerned about toxin manifesting differently for them. So lots of different things to think about through the whole life of one of these animals.
So look out for Alicia's future research on this, because it's obviously something she's delving into.
Watch this space. Yes.
Okay. Move on to our next questions from Valentina Melika. Thank you for the presentation. Very interesting research. I have 2 questions. 1, did you see any difference between gastric content samples and colon content samples? And I'll jump to the second one in case it's related. Did you find any difference among different demographic groups, male, female, juvenile, and adults? Thank you. Yeah, so between gastric content and colon, and then were there differences among different demographic groups within that?
Great, love it. So we have this table from our paper, and I apologize, tables are probably the most boring way to look at data. But these represent our individual seals from which we had both stomach content and colon content samples that were taken at the same time. And we analyzed them all for DA and saxitoxin, and you can see that colon content is regularly has a higher toxin concentration than stomach content. That's particularly true in saxitoxin here. These BDLs mean below detection limits. So essentially all of these stomach contents did not have detectable saxitoxin, while several of the corresponding colon contents did have detectable toxin levels. And this is probably multifactorial. So you first thinking about as material moves through the GI tract, you're removing water. So it kind of makes sense that things could concentrate the further you get. It is also the case that colon contents will often represent more than one stomach contents of food. So if a stomach content will look at your last meal, your colon content could encompass multiple last meals. So it also kind of makes sense that toxin concentrations could be higher there. This is actually exactly why we restricted to our comparisons when we looked temporally or when we looked between regions to comparing the same type of samples. So we didn't look at stomach content versus fecal content. If one location had mostly fecal content and one location had mostly stomach contents, we didn't compare between them because those wouldn't be appropriate. The second question, which you're gonna prompt for me right now, hopefully.
I will. So the first question was, did you see any difference between gastric content samples and colon content samples? Two, did you find any differences among different demographic groups?
Yes. So great second question. We, in the same way that we had sort of even age spreads between our different seals, we did also have about a 50-50 male-female split. And very early on, I looked at some initial comparisons, just like do males and females overall look different? And I did not see trends. I didn't do a comprehensive analysis of that breakdown, mostly because the information that we had for our samples was a little bit different. These were samples coming in from lots of different places. So again, because these were opportunistic, they weren't perfect for doing those kinds of comparisons. But on a sort of cursory look, I didn't see big differences on those demographic factors. It would be worth following up when we get some more comprehensive data moving forward. There's ongoing research looking at sort of regularly sampling now that we've seen some initial interesting results. Yeah.
Okay, thank you. That is the last of the questions from the Zoom. Daryl, do you have anything?
Yeah, I've got at least one more question I might ask you, and that has to do with the impact of these toxins on the lower level organisms that are being consumed by the seals, and to what extent do these end up in major die-offs of these lower organisms, and is the sort of level of toxin consumed by the lower organisms a determinant of whether or not they're clinical levels for them?
Yeah, this is a great question. So because these toxins sort of initially showed up for us as human poisonings, a lot of the work has been looking at humans and other higher level trophic organisms. But because, as you identified, they move up through a food web, there are all kinds of organisms also being impacted by or also interacting with these toxins on the way. And to my knowledge, research into sort of impacts on shellfish and things like that don't show a lot of impacts of the toxin. I suspect that that has to do with the fact that these toxins are interrupting processes in the brain, and those manifest very differently depending on the different organisms. There was some initial work that was looking at, I don't know the stage that it's in, but was looking at possible impacts of toxin on fish and specifically fish activity. And I haven't seen a sort of latest on that. So that to me is an interesting thing to look into. People have been mostly concerned with animals having sort of overt die-off or stranding events, and those are the higher order organisms like seals, otters, walruses, that kind of thing.
Okay, thank you. Eric, I guess we have some additional questions, or are we done?
Looks like just comments and
Just
high
comments.
praise for Alicia. Excellent presentation answers, and lots of thanks for your answers on the questions.
Thank you to
Okay.
everybody who asked the questions. It's always great talking to audiences and hearing how different things that we're doing are landing.
Well, again, thank you, Alicia, for a great talk. I look forward to seeing more papers from your PhD work published. Maybe they'll be in Marine Mammal Science. We'll watch for them if they come our way. Thanks for taking the time to do the presentation tonight.
Thanks so much for having me.
You're welcome. I guess on that note, we're right at 8 o'clock. We've had a good session of question and answer at the end. We'll call it an evening. All right. Thank you much. Good night, everyone.